Well, hello, folks. Hello, and welcome to the Apologetics Academy, a live interactive webinar I run on Saturdays. We bring on different scholars, apologists, thinkers, scientists, philosophers from across the theological, philosophical spectrum to talk to us on subjects of interest to Christians, especially on topics pertaining to whether Christianity is true. And we also cross-examine and evaluate other worldviews as well, such as Islam, Mormonism, etc. And the format is we have a presentation by a special guest speaker, and then we open up to open floor Q&A discussion and dialogue. There's a number of ways you can participate. There's a chat window. You can participate in the chat with other guests. You can also click the raise hand button, and when we get to the Q&A, I'll promote you to a panelist and have opportunity to interact with the speaker. And you can also click the Q&A button at the top of your screen and submit a textual question that way as well. So let me just introduce our guest tonight, or today. We have Dr. Paul Nelson, who is a philosopher of biology, who's affiliated with the Discovery Institute Center for Science and Culture in Seattle, Washington, and he's also on the faculty with Phyla as well. So without further ado, let me just turn it over to Dr. Nelson. We're going to be talking about ontogenetic depth and why it matters for evolutionary theory. So Dr. Nelson, take it away.
Thank you, Jonathan. It's wonderful to be back here at one of the liveliest intellectual sites on the web. I have to apologize at the start that the presentation today, and you'll see from my title slide, this is part one. Part two deals with proposed solutions to the problem of ontogenetic depth. This presentation will have relatively little directly to do with apologetics. And I hope that doesn't disappoint anyone. In part two, I hope to talk more about how intelligent design plays a role in understanding the patterns I'm going to describe. But I suppose the people who tune in to the Apologetics Academy are accustomed to ranging widely over intellectual subjects, and they will perceive the relevance of this topic to their own interests. At least I hope they will. All right, let's get started. I have a lot of ground to cover, and for that reason, in fact, I split this talk into two, and Jonathan and I will be scheduling the second part as soon as possible. This topic of ontogenetic depth has caused me considerable pain over the last 14 years. Very briefly, summarizing a fairly long history, I proposed an idea in a poster at the Society for Developmental Biology, I think in 2003, and it crashed and burned more or less immediately. And then, as many of you probably know, P.Z. Myers has, on the basis of some hasty promises that I made that I couldn't fulfill, P.Z. Myers has held my feet to the fire annually ever since. And I have to say I'm grateful for that. And I'll later in the presentation talk about why I'm grateful, despite the pain. But I've continued to think about the topic, and in fact, I've had lots of conversations with people about it. So this phrase you see here in quotation marks is an interlocutor of mine telling me, oh, come on, Paul. Your title slide is a non sequitur. So I'm imagining myself with a venti-sized coffee in Starbucks sitting across the table from someone like PZ or friends from University of Chicago, let's say, and they would say, look, just stop right now. You can stop the presentation right now. Your title slide gives it away. You say there's this property in animals, and by the way, I will be talking only about animals today, not about higher plants, although my guess is the phenomena I'll be describing of tissue differentiation, cell differentiation, increase in cell number would apply, especially in the angiosperms as well. I just haven't looked at any of the relevant data or literature. So I'm talking just about the animals, the metazoa today. Anyway, my friend in Starbucks says, look, you say there's this property in developing animals, but you can't define it or measure it. And any concept that's so nebulous can't possibly be of value to biology. All right, the point of the presentation today, and part two as well, is to show actually it's real. Ontogenetic depth is real, and I hope to establish that in the next hour. But secondly, and I think more importantly for evolutionary theory, the problem of constructing ontogenetic depth via mutation and selection is closely related to, indeed it may lie at the heart of the unsolved evolutionary puzzle of the origin of animal body plans. So those are the two propositions I want to establish in the presentation today. And then of course I want to hear from you in the Q&A. So in one sense, ontogenetic depth is just completely obvious, all right, because it's trivially easy to define as a distance. This is very close to what I had in the Society for Developmental Biology poster. It's not word for word, but it's very close. Ontogenetic depth is the distance for any animal, any metazoan, between a single-celled state and the adult capable of reproduction. Okay, so we're talking about an interval here between a fertilized egg. I'll talk only about sexually reproducing, or that is a species that produce gametes, eggs and sperm, and the interval that we're talking about here is between the single-celled state, the fertilized egg, and the adult capable of reproduction, okay? Now, these distances really exist, right? I didn't make them up. You can, in fact, you can watch them. So here is from a nice video from the Bob Goldstein lab at UNC. This is a C. elegans embryo. We're actually starting here at the 2-cell stage. So there's already been one round of cell division. Here is the clear egg capsule surrounding the zygote, or now the embryo. So let's just get this running. Up here you will see the time. This is the real time here. I think it'll be about 14 hours if I remember correctly, compressed to about 45 seconds. Now, John Sulston, who died this year, and I regret that I never had an opportunity to meet him, because when I was a student, his beautiful cell lineage of this species was published in Developmental Biology and really shook up a lot of people. For one thing, the lineage looked completely illogical, not the sort of thing we would design, but it was a case or an episode of year after year after year of meticulous observation with special microscopy to map what you're about to see. So I hope this will run for you. So these cells are going to begin to divide. The total cell mass will stay within the egg and will go to the point of hatching. All right, let me just kick this off. So these cell lines are dividing and specializing as they do. Now you'll notice there's a great deal of movement within the egg capsule. That will be relevant later on and something that we're going to talk about, I think, roughly halfway into the presentation. Here now you can begin to see the form of the worm emerging, and there's a lot of movement there. What's going on as we watch this is cell number is increasing from 1 to about, at hatching, I think it's about 580 if I remember correctly. The adult, the reproductively capable adult, will have about 1,000 cells. So this is now at the point of hatching. And watching this year after year, recording which cells gave rise to which, is what won John Sulston the Nobel Prize in Medicine and Physiology. Just a beautiful piece of observational biology. So there's our total time elapsed. Nearly 15 hours compressed into 45 seconds. That what you just watched is a bona fide biological distance. You're going from a fertilized egg to the, I think, roughly 580, if I remember correctly, cells present at this point. The hatching occurs, and then cell number continues to increase, mostly in the reproductive tissues of the worm. That's a real distance, okay? Now, you can illustrate it this way. Here's our starting point. Here is cell number and differentiation increasing until we see the form of the adult reproductively capable worm. So here's my definition over here. Now, the definition describes a distance that exists whether I say anything about it or not. It's out there in nature to be assessed. Okay, that's the obvious sense of ontogenetic depth. In another sense, however, it's not obvious at all. How do you measure that distance? Okay, and P.Z. Myers and others have pointed out there's little grounds for saying that a distance exists if you cannot specify the units of that distance, right? As I'll discuss in a moment, the distance from our planet to the sun can be given in meters or miles, or you just name that interval the astronomical unit as it's now designated. But you can't sensibly talk about a distance without giving some units that anyone then could use to determine either that distance itself or to use those units in some other species to look at their ontogenetic depth. So right from the start, when P.Z. Myers looked at this ontogenetic depth proposal, he said, well, what are the relevant units? If you don't know what those are, you can't possibly measure ontogenetic depth. So at the time, 2003, 2004, I had suggested that the distance could be measured in terms of cell type and cell division. So that's what's happening here as you have this expanding tree of developmental decisions. You've got, and we'll see later in the talk, the more detailed diagram of the C. elegans cell lineage. You have cell number going up, but you also have cell types arising: reproductive, cuticle, gut, nervous system, and so forth. You have differentiation occurring. So my very primitive and incorrect approach to this was to say, well, you could get a metric units from those two criteria, or those two aspects of C. elegans, namely cell division and cell type. And that would give you a metric then for assessing ontogenetic depth from here to there, okay? Now, the problem is even worse though. I'll come back to those two I don't want to call them criteria, two properties, cell type and cell division, later. But I want to say the problem is even worse. And in that sense, P.Z. sort of underplayed just how bad my initial proposal was. So let's go back to my astronomy example. There's the sun keeping us warm, although this spring I've wondered about perhaps the sun is getting lazy or taking time off. It's been the coldest spring in my memory. Here in Chicago, it's been the coldest spring since the late 19th century. In any case, we can name that distance. So here we are, we look out there, we see the sun, we say, well, how far away is that? Well, we can say, well, we can come up with something in terms of feet or meters or some other unit to assess that distance. We can name that distance, call it the astronomical unit if you like. Measuring it, however, is a very different matter indeed. And in fact, today the distance is determined by radar and other direct methods. But prior to the invention of those technologies, you had to do trigonometry, right? And to do the trigonometry accurately required observing the transit of Venus across the face of the sun from two positions as widely far apart as possible on this planet so that you can determine the angles that you would need to make the trigonometric calculation. That was very hard to do. And in fact, determining the distance to the sun on the basis of parallax and trigonometry was really did not settle down to the value of 92 million miles until fairly late in the development of astronomy. Aristarchus, a brilliant Greek mathematician of about 300 BCE, got it wrong. He knew that he could use the regularities, mathematical regularities within a triangle to predict, or not predict, to calculate the distance to the sun, but he got it wrong. He was off significantly. So there's a difference, an important difference between naming a distance and being able to measure it. So the fact is, even if we could specify the relevant units, it's unclear that we could reliably measure anything with them. That is with respect to ontogenetic depth. So back in the Starbucks, and my friend sitting there is staring at me, and he says, "Paul, you've dragged us to slide 12, and ontogenetic depth is still DOA." Well, not really. Now, if you don't remember anything else about this presentation, this is a take-home that you should remember. The fact that we cannot measure a distance does not make that distance go away. Let me repeat that. Well, I'll go back to determining the distance to the sun. How far away is it, right? We can name that interval, give it a name, Earth to sun distance. Who knows, right? The answer in this case lay centuries in the future, very, very late in the development of observational astronomy. Yet the distance is still there, and it won't go away because you can't measure it. Come back to biology. The fact that we cannot yet measure ontogenetic depth does not make the distance that we're going to be talking about go away. It's still there. It exists. This property of developing organisms exists and must be explained by evolutionary theory because of the conjunction of a pattern theory, a historical pattern, and a process claim. All right? So as is often the case in evolutionary theory, Darwin sets the puzzle for us with the following conjunction. We have a pattern, namely the common descent of the animals from a unicellular eukaryotic ancestor. So that's our historical pattern of relationship that we've hypothesized represents the actual topology or history of the animals on this planet. Then we have a process claim. Natural selection was the main cause of biological novelty, including the origin of animal body plans. Now, when these two are conjoined, the problem of ontogenetic depth emerges quite readily, as you'll see. So we're going to spend the rest of this presentation before the Q&A dealing with how these two parts of evolutionary theory generate the problem of ontogenetic depth. Okay, so let's start with the theory of historical pattern. Get that on the table first. Here is a phylogeny of the Metazoa, of the animals. And by the way, anyone who wants this PowerPoint or any of the references, please contact me. Jonathan has my email, and if you write to him, he'll pass it on to me, or perhaps I can find a way to just give you my email directly. Some of these images, I think I'm safely under fair use, but some of these images are copyrighted. So if I give you the PowerPoint, you cannot put it up in any forum that would fall outside fair use. All right, I just need to stress that. But please consider the PowerPoint is if you want to look at it in greater detail, I'll send it to you, and I'll send you any of the references as well. So here's a phylogeny of the animals. We'll talk more about C. elegans. Here they are up here with the nematodes. Now, you don't need to worry about the details of this phylogeny. Any such phylogeny of the Metazoan phyla is very controversial, and there are some interesting hypotheses here. You have the origin, for instance, of segmentation here at the protostome, deuterostome, common ancestor, and then that character is lost repeatedly along these branches. That's what this open red rectangle indicates in these cases and so forth. There are other losses too. But what I want to focus on is just a portion of this phylogeny. In particular, it's not represented here, but the outgroup for this, this phylogeny is a single-celled eukaryote known as a choanoflagellate. So here's a beautiful photomicrograph from a guy in Germany. The name comes from, choano is Greek for funnel. So you have this funnel-shaped collar here, and then the flagella arise from within that collar, within that funnel-like collar. These look very much like the feeding cells of sponges, and it's interesting to ask, are these sponge cells that went rogue and said, "We're not, we don't want to be sponges anymore. We're going to go off and work on our own." And I make that as a joke, but it's actually been entertained as a serious hypothesis for the origin of choanoflagellates, although it's not the main view now. In any case, this is the outgroup that's not represented here in the diagram, but would be branching off at this point. What I need you to keep in mind here is something akin to a choanoflagellate. A single-celled eukaryote lies at the root of the branching pattern for the metazoa. So here I've taken just a portion of that previous phylogeny. We have choanoflagellates here. This is a different photomicrograph lit up by some fluorescent markers. Here are placozoa. Actually, this lonely species, Trichoplax, is the only child in this village. This is a phylum with a single species in it. Very, I think, 4 or 5 cell types, not much differentiation here. Fascinating creature. You can sometimes observe them if you're lucky on the wall of an aquarium where they cling. Sponges and C. elegans, which we'll come back to, okay? Now, the dotted line here indicates that there's a great deal more branching to other groups that I haven't represented. But I wanted to focus on nematodes in particular, comparing them to choanoflagellates and other single-celled eukaryotes, okay? By the way, something very interesting to keep in mind for part 2 of this story. Placozoa have had their DNA mapped, or Trichoplax has had its DNA mapped. And it turns out that many of the sophisticated regulatory elements that you see in, let's say, in arthropods or in other groups, much fancier than Trichoplax, are present in Trichoplax. So here's how Charles Marshall and James Valentine put it. I'll come back to this in part 2 because it's really interesting. They put it this way. They say placozoans contain genes that in cnidarians or bilaterians are involved in nerve cell specification. So notice the tissue type here. Muscle cell specification and circulatory system development, and evidently in anterior-posterior patterning, head-to-tail patterning. Yet, placozoans have no nerves, no muscles, no circulatory system, and lack an AP axis. It's from a paper they published in 2010, a couple of years after the Trichoplax genome was examined. Now, this is a really interesting puzzle because many of these genes are also present in choanoflagellates, single-celled eukaryotes that don't go through anything like cell division and differentiation that we see in the metazoa. This puzzle is world-class, and I'll come back to it in part 2 of the story, but I just wanted to indicate to you that it's there and it's very interesting, and there is a backstory involved with this that is worth telling. I just don't have time today. But keep that in mind, and it's an advertisement for coming back for part 2. All right, so What's the last common ancestor down here? Well, we can put the branch in, right, or the dotted line indicating that there should be a branch. So the last common ancestor of these taxa was a single-celled eukaryote, probably something like a choanoflagellate, okay? Now, if you look at cell number in an adult nematode, it's roughly 1,000. They're not very big creatures, right? About a millimeter and a half long, but these cells are specialized into tissues, and you can see you have a very distinctive, it's elegant actually, hence the name, elegant morphology for this worm. And those tissues are performing particular roles, right? You've got gut, and as I said a moment ago, cuticle, nervous system, and so forth. In particular, for the mechanism of natural selection, as we'll come to in a moment, you have gametes, eggs and sperm in the hermaphrodite form of C. elegans. So what's going to happen along this branch? Well, two things. Cell number is going to go up, and degree of differentiation is going to increase dramatically, okay? Now, it's net gain, okay? It's net gain. So you could have had a more complicated ancestor to C. elegans, and that ancestral, somewhere along this branch, that ancestral form lost cells and lost differentiation. But when you get to the end here, to the terminus, where you're looking at a species around today, total count is going to go up. Cell count and again, degree of differentiation is going to increase. So that's going to happen. It has to happen, in fact, along this branch, if that branch actually existed. All right, so the specific developmental pathway that we observe today in C. elegans must have been built incrementally over evolutionary time, starting from a single-celled eukaryotic ancestor here. Oops, I'm sorry. Along this pathway, all right, that evolutionary lineage. Okay, here's another way of representing it. This is a ciliated protozoan. It's a single-celled eukaryote. Single-celled ciliated protozoa are weird in so many different ways. I mean, they're incredibly sophisticated in so many different ways that it's really not a good model for an early arising eukaryote leading to the metazoa. But, right, it's PowerPoint, right? And I got to put the slides together. So it is single-celled, but please don't take this as my hypothesis for a good example of the ancestral form. It's just there to hold a place. What's happening here? Well, along that evolutionary branch that I showed you in the phylogeny, cell number has got to go up. And it can't go up all at once, right? You're going to be adding cells along that pathway. Cell number is going to increase, but that's not the only thing again. Specialization, cell specialization will be occurring. Now, ontogenetic depth, when you start to disarticulate the concept, actually entails two causally related dimensions. There's the developmental pathway that we observed today. We watched the video from the Goldstein lab of that happening. And there is the historical or evolutionary lineage that we can't see, but we infer, that constructed that pathway where it did not exist before. And these two dimensions turn out, and this will be my punchline for this presentation, these two dimensions turn out to be in significant tension with each other, especially if the causal process you're invoking to build one, right, is natural selection. All right. So let's turn to natural selection, all right? The main process hypothesis in play. Now, probably some of the people watching today, I hope, watching anyway, are not too keen on natural selection, right? I know that when several of us from Discovery Institute were in London in November 2016, many of the people in that auditorium at the Royal Society, such as Larry Moran from Toronto, have a rather low regard for the causal efficacy of natural selection as an evolutionary process. However, I should point out that they tend to be in the minority with respect to the total community of evolutionary biologists. So Douglas Fatuma is a very prominent evolutionary biologist at SUNY Stony Brook. And in this paper 3 years ago, he said, look, existing theory, textbook neo-Darwinian theory provides a plausible account for most or all evolutionary phenomena. He says, I don't know of any macroevolutionary phenomena that are inconsistent with existing theory, or that would require us to reject one of its principles as simply false. Well, right at the top of the list of those, of the causal processes embodied in existing theory is natural selection, all right? So Dawkins, who is always very clear and straightforward, I love that about him, he says, look, natural selection is, well, this is my paraphrase of Dawkins, natural selection is both necessary and sufficient to explain adaptive complexity. Or as he put it in the early 1980s, the theory of natural selection provides a mechanistic causal account of how living things came to look as if they had been designed for a purpose. Now, you might say, Paul, that's Dawkins, for goodness sake, you're quoting him from the early 80s, right? Well over 30 years ago. Actually, come right up to the present, the causal primacy of natural selection remains the dominant view within evolutionary theory today. So here's a paper published actually in response to the agitation at the Royal Society by Deborah and Brian Charlesworth and Nicholas Barton, published last year. And they say, we finish by re-emphasizing the central concept of neo-Darwinism and the modern synthesis, namely allele frequency change caused by natural selection is the only credible process underlying the evolution of adaptive organismal traits. So leading up to this point in their paper, they survey some of the various alternative processes proposed at the Royal Society, such as niche construction, Jim Shapiro's adaptive genetic engineering, gosh, I don't remember all of them, but they went through them one after another and pointed out that those processes, which may play some role in evolutionary change, are insufficient to explain adaptive organismal traits. So they're focusing on that. Well, if anything is an adaptive organismal trait, it's a metazoan body plan, all right? So here is natural selection. I should go through this a little more slowly. Just as a refresher, I know you guys probably have all been through this, but it's worthwhile just taking it apart very carefully. Expressed as a conditional, and this formulation actually does not suffer from the problems with circularity or vacuity that sometimes affect formulations of natural selection that employ the concept of fitness. So this formulation, as you'll see, does not use the concept of fitness at all. It's been eliminated. You got a conditional if within a species or population the individuals vary, all right, in some trait. Leave different numbers of offspring in consistent relation to the presence or absence of trait Q. And consistent is important because if the sign is varying, that's drift by definition. So there has to be a consistent relation between the presence or absence of Q and the number of offspring produced. And lastly, transmit trait Q faithfully between parents and offspring, the condition of heredity. Then the frequency of Q will differ predictively between the population of all parents and the population of all offspring. And this formulation is the work of John Endler and Richard Lewontin. And they have meticulously set aside the concept of fitness, which causes a great deal of mischief in other formulations of natural selection. All right, this works, okay? And it's a real process. Just as a reminder, we've got 3 necessary conditions, which, if satisfied, will give us selection. Now, as I said a moment ago, many people, many evolutionary biologists in particular, are skeptical of the necessity or sufficiency of selection. And they say, look, it's overrated as an evolutionary cause. You've got to look elsewhere. I have in mind people like Michael Lynch, for instance, at Indiana, who's written very critically of the excessive reliance on selection in evolutionary theory. But here, look, any evolutionary process, drift, self-organization, pick your favorite process, must satisfy the following. All right, so we can generalize what I'm going to say in the second part of this talk to any evolutionary process. It doesn't have to be selection. You have to have differences, right? You've got to have novel variations. Without novel variations, this planet should be swarming with trillions of species of, I don't know, cyanobacteria or whatever, right? There wouldn't be blue whales and Homo sapiens and oak trees and all the interesting stuff. You've got to have differences. Those variations must make a difference. Behavior, phenotype, physiology, anatomy. If you want to explain the diversity of life on this planet, the variations have to make a difference to the organism in question. And the difference has to be permanent, right? The genetic basis of that difference must be capable of transmission to the next generation, stable transmission. It has to be a permanent change. One of the criticisms that the Charlesworths and Barton made, for instance, of Avia Blanca's ideas on what we might call neo-Lamarckian inheritance is it's not stable. It doesn't hang around, okay? You've got to vary. Leg one of the stool. Two, those variations have to make a difference to you as an organism, a difference that would be detected by an observer, let's say. And you've got to pass the difference on for keeps. All right, keep this in mind, because the critical analysis that I'll be making of selection with natural selection with respect to ontogenetic depth actually applies to any evolutionary process that you care to name. They all have to fulfill, they all have to satisfy these requirements in order for there to be any kind of directional change among organisms, okay? So this slide just repeats what I said. The toy and real examples I will present next apply to any candidate evolutionary process, not just natural selection. Okay, now let me go back to my naive way of thinking about this that PZ nailed me on. I said, well, look, you know, if you had cell division and cell type, you could get a simple metric that ought to apply across any Metazoan group, because they all undergo cell division and they all have a diversity of cell types. And that naive conception comes from looking at something like this. So this is what John Sulston won the Nobel Prize for, working out this cell lineage, which I think, if I remember rightly, is what's represented here at the terminals of these various branches is what's present at hatching. And the cell number will continue to increase, but this is the main feature. This is what happens in any wild type C. elegans. Starting here at the zygote, and then you've got these branching lineages set up where the terminals are going to have specialized functional roles within the worm. So I look at this as a somewhat naive biologist trying to come up with a simple metric, and I say, well, just count up cell types and take the average number of cell divisions for any of these lineages starting at the zygote and going down to a terminal, which is, in the case of C. elegans, 7 to 9, right? There's your metric. Ontogenetic depth would then be equal to the average number of cell divisions between the zygote and any terminal cell multiplied by the number of different cell types in the termini, okay? So assuming we can track cell lineages, and as I recall, PZ said, well, there's a big question mark attached to that. And by the way, in part 2, between my naive proposal in 2003, 2004, and today, there have been a number of amazing breakthroughs on tracking cell lineages that I'll talk about in part 2 that are really, really interesting. In fact, something just published in Science, the very latest issue of Science, a project looking at flatworms, that it's just amazing. But I said, right, let's just count cell divisions, we'll look at cell lineages, and then we'll multiply that by the number of distinct cell types in the reproductively capable adult, and that gives us ontogenetic depth. It shouldn't be too hard to do. Wrong. And again, I want to thank PZ for noticing this. It's always best to kill a bad idea as soon as possible, and he performed that service for me by pointing out there were problems with this conception. But we can see those, and I was so, I mean, it was embarrassing, to go back and look at what I had suggested and then think about it for a minute and realize it wouldn't work at all, okay? So here's a toy example. My initial proposal, that is, wouldn't work at all. Here's an egg. I don't know if you guys can see that on your screen. If you can see my cursor. Here's our starting point for this little toy organism alpha. And it goes through something very like a canonical cell lineage that we see in C. elegans. And we have 5 cell types represented at the terminals and an average of 4 divisions between the starting point in any terminal cell. So 5 times 4 is 20. Ontogenetic depth, 20. Now notice the adult phenotype, because after all, this is not what the organism is going to look like. This is a cell lineage diagram, okay? Our adult phenotype might have this form, where let's say this is the, hang on a sec. Yeah, let's say this is the germline, the cell here, the red cell here. It's going to, do it all again in the next round of reproduction. This phenotype is not the same as this phenotype. Here, the germline is moved from the position it was in the previous slide. Toy's organism alpha moved to an interior position. You've got this big central cavity here, yet the ontogenetic depth of these two critters, these two toy critters, is exactly the same. It comes out as 20 in both cases. And these lineages are exactly the same. So what's missing is the whole question relevant to Metazoan body plans, which is form. Form is nowhere present here, right? If you think back to that video, I won't go back and run it again, but if you think back to that video, and I'll talk more about this in part 2, there is a great deal of motion going on in those cell lineages. Those cells are scooting all around because they have to get in the right positions, not just with respect to themselves, right, but with respect to everybody else that's going to be present in the adult phenotype. There's a great deal of movement going on, and that's not represented here anywhere. And there's no way to take account of it if all you're doing is counting cell divisions and cell types present at the terminals. So it couldn't possibly work as a decent metric for assessing Metazoan developmental complexity. Right? It's just a failure right out of the box. So this, if you look at this diagram, you're getting a lot of interesting information, okay? But that is not that. And this is what we need to explain. This elegant tapering shape where cells are in the right place with what they need to do, with respect to what they need to do for this worm to make a living. So, I read PZ's critique and I think, Paul, you really blew it. This is just not the same as that. And you have no way of accounting for cell movement, 3-dimensional, actually 4-dimensional when you add the element of time into your analysis. The story is vastly more complicated, all right? Now, big worry. It's not a worry for you, it's a worry for me, because I'm very interested in pursuing ontogenetic depth towards the goal of coming up with some kind of objective metric that could be applied among different metazoan groups. I'm still interested in that. But almost nothing of any genuine functional interest in biology can be represented by a single number. Really, it's one of the problems with the concept of fitness, that it reduces the complexity of organisms to the interval between 0 and 1. There's got to be something wrong with that, right? Laying aside whether you can assess fitness or not independently of reproductive output, reducing the complexity of organisms to a single number between 0 and 1, there's got to be something wrong with that. Well, I'm worried that any metric that will issue in a single number is bound to be missing something. So this is I ought to print this slide out and post it here in my office just to remind me, don't try to do something that can't be done. But let's go on. All right, why does natural selection fail to explain the origin of metazoan cell lineages and differentiation? Okay, let's go back to our formulation of selection. The condition of heredity, is going to turn out to be critical. Remember, you've got to have all three of these satisfied for selection to operate. If any one of them is missing, there's no selection. So keep this in mind. Pass it on for keeps is going to end up being the critical question when we try to evolve cell lineages with differentiation from single-celled ancestors. All right, so let's start with some toy examples, and then I'll move to some real biology once we have the toy examples in place. Look, if natural selection does not work even in these toy examples, it won't work with more complicated cases, vastly more complicated cases, such as real animals. So I got to speed up if I'm going to finish on time for a Q&A. I think we'll make it, though. All right. Let's take the simplest possible case. All right. We're going to boil it down to the simplest possible case to run our analysis. So let's start with what we could call cell 0. And cell 0 is going to divide into 2 daughters. Okay. Now we're going to put instructions in place. Never mind about the genetic details. Just think of these as instructions in a game where you want the entities in the game to perform certain actions in the right sequence, but they have to be instructed to do that. So cell 0 is going to divide and produce 2 daughters, all right? They have to stick together. They can't go their separate ways. This isn't yeast where under normal conditions, unless you've been hitting them with mutations that cause them to stick together, yeast under normal circumstances divide, and you get 2 separate daughters. Now we say to these cells, hang around. You've got to stick together if we're going to build an animal. So the first instruction we put in place, first instruction we put in place here is stick together. That's present, it's got to be present here in cell 0 for this to happen in the right sequence. But of course, that's not enough. The daughters must differ. Now, this is critical. If the daughters don't differ, what you end up with, or what you would end up with if cell division continued, is a clonal mass. And a clonal mass is not an organism, or a clonal mass is not a metazoan, all right? So this would be bad if you're trying to build an animal, because the daughters are identical. They're sticking together, yes, that's interesting, but if that's all they're doing, you're not going to get what you need, namely differentiation to build different structures and tissues and so forth, all right? So that won't work. Simply multiplying cell number without also differentiating at the same time will take these cells in the wrong direction if what you want to build is an animal or a novel body plan, all right? Now, instruction 2 comes into play. The daughters must differ. Sorry, this is skipping a little bit. We put instruction 2 here, all right? But there is also instruction 3. And that is there because we have to satisfy this. You've got to pass it on for keeps. So all of these novel variations, if we're going to get them into the next generation, have got to be inherited. They've got to make it through reproduction. Okay, so what's happened in this case is the daughters are differing. That's good. We want to be able to do this, however. We want to get this same pattern again, because that's what natural selection is going to operate on. So we need a third instruction. One lineage has to remember all the instructions. One of these lineages has got to play the role of the record keeper, if you will. That lineage which retains all these instructions to make these divisions. That will be the gamete, so to speak, that's going to come down here and start it all over again. Now look what we've done. We've loaded cell 0 with multiple independent instructions. Divide and stick together. Daughter cells must differ. One lineage remembers all the instructions. Natural selection is nowhere to be seen because reproduction actually hasn't happened yet. Okay? One of the necessary conditions of natural selection is the reproductive cycle. You've got to pass your genes on so that their functionality can be assessed by the environment and so forth. That's the only way you're going to get differential reproduction. There is where natural selection begins to operate, at that blue star. Only after reproduction occurs does natural selection come online, so to speak, as a causal process, which means that all of these instructions have to be preloaded into cell 0. And there's just no way around this if natural selection is going to be operating to give you increase in cell number and diversifying lineages. It's part of the logic of the process itself. And this, as far as I can tell, maybe there's a simpler toy example. This is the simplest toy example I can conceive. And you'll see that natural selection does nothing here. Not until you've actually built cell 0, instructed it what it needs to do, and get this to happen, does natural selection begin to operate. All right, let's look at a real life example. Go back to C. elegans. Here is the early cell lineage of that worm. Very first branching pattern. So we're going to focus on this part right here. All right. Notice that the first division and those to follow set up asymmetrical daughters. Okay, there has to be an instruction here, wherever it is in C. elegans, in the DNA, who knows? There's got to be an instruction here to say, you're going to be this daughter and you're going to be this daughter. This is the germline coming off here on the right. At this division, there has to be an instruction because these daughters are asymmetric. They're not equivalent and so forth. There has to be an instruction here. Okay? So this early cell lineage is a defining character of C. elegans. All right? It has to be explained. Somewhere along that branching path from single-celled eukaryote to C. elegans, this character arose. Now the question is, when? When along that branch did it arise? You can see here, these daughter cells are going to be the progenitors for, in this case, germline, muscle, gut here with cell E and so forth, all the way out to the roughly 1,000 cells of the adult worm. All right, when did this character evolve? That's the question we're going to ask. Now, we can go back to my diagram earlier, and again, I want to provide the caveat that I don't think a ciliated protozoan is a good model for the ancestral eukaryote, but never mind that. It's just a cartoon or just a figure to hold a place there. Somewhere along this lineage, that character, this one, came to be. And the question is where? Now, this ends up being very, very tricky to answer. And one of the things I'm going to talk about in part 2, and it was quite a shock to me to discover this, when you go into the literature, the Evo-Devo literature, going back decades, I'll talk about this in some detail, There is almost no literature anywhere on the origin of cell differentiation. It's held up as an unsolved problem, but I'll tell you, it's been an unsolved problem for a long time. And I think the reason for that is natural selection as a causal process is in principle insufficient to explain it. So come back for part 2 when I'll give you the Evo-Devo backstory, but there's almost no relevant literature dealing with this question. Okay, well, this stands for C. elegans initial cleavage pattern. That's what that little acronym stands for, okay? Did it evolve here? Now remember, if we place the evolution of that character here fairly early along this evolutionary trajectory, none of this exists, all right? It's yet to evolve. Cell number's not high enough yet that you can build these tissues. So you've got some very, very small ancestral form with a relatively small number of cells, perhaps around 150, 200, somewhere in that region. This character evolves. None of this exists yet. So whatever this does now for C. elegans, it couldn't possibly have been doing when it evolved here, because the downstream cell lineages that it's going to specify don't exist. What that means is that you actually can't describe this character at the point that it evolved. Because, go back here, these decisions that are being made with respect to their distant downstream consequences. That's the actual function of this early cell lineages, is to set up these daughter lineages to head off, to proliferate. The cells are going to proliferate with the right functional roles in that adult worm. That's what this is doing. That's what this character is doing. But none of that exists. So you actually can't describe this character when it first appears because it's not doing the job that it does today, which means your description of the evolution of the character is going to be completely bloodless. Vacuous, actually. There won't be any content to the explanation because this, hate to say it, right? It's teleological with a vengeance. This is an end-directed character. And there's no point in having an end-directed character if you don't have the ends for it to direct. All right? Let's move it. Let's move the point of origin later. Cell number now is up 700, 750. Could it have evolved at that point along that branch? Well, what's back here? This is a clonal mass. There's no differentiation going on because the character that sets up differentiation hasn't evolved yet. So again, you will be unable to describe the ancestor to C. elegans with any specificity. It's just going to be a clonal mass, and differentiation itself will not arise because the character that provides that is yet to evolve. And it really doesn't matter where along this trajectory you move. It doesn't really matter where along this trajectory you move this character, you're going to run into problems. Either you've got a clonal mass with no differentiation, or you've got a character that's specifying things that don't exist. And if natural selection lacks anything, it lacks foresight. There's no foresight with natural selection, or really any evolutionary process that you care to name. This is why I said earlier that the critical analysis that I'm doing here really applies to any evolutionary process, drift, self-organization. Tick through your favorite list of alternatives to natural selection, the same difficulty is going to apply. All right, I need to stop. Take homes, and then I'd love to get your questions. And remember, some of what you may be asking will be dealt with in part 2. So if I get a question like that, I may defer it to part 2. Not because I'm being coy, but because we didn't talk about what's going on in Evo-Devo. And we didn't talk about a lot of the dimensions of this problem that I'll be discussing in greater depth next time. Ontogenetic depth is real. Whether you can measure it or not, it's out there, and it has to be explained by evolution. And the problem of constructing ontogenetic depth via mutation and selection, or any other evolutionary process, is closely related to the unsolved evolutionary puzzle of the origin of animal body plans. At its heart, the problem has to do with what development is, what animal development is. Animal development is end-directed. And if animal development is end-directed, you have to find a way to build that process where you don't know where you're going. That is going to be a problem for any undirected evolutionary cause. All right, I will stop there, and I would love to have your questions.
All right, well, thank you, Dr. Nelson. Do you want to unshare your screen at this point? Just click the...
I'm sorry?
You want to unshare your screen at this point? There you go. Great.
Can I go back to my slides, though, if
Oh, yeah, sure. You can.
Questions arise?
Yeah, sure. Absolutely. Folks, this is the interactive portion of the program. If you'd like to ask a question for Paul Nelson, then simply raise hand by clicking the raise hand button at the top of your screen, or the Q&A button to submit it that way. Also, you can also submit it on Facebook if you're following the Facebook live stream. Glenn Gardner submitted a question saying, why is population almost always a big part of these discussions when heritability can only occur at one reproductive event at a time?
I'm sorry, Jonathan, could you repeat the question? I didn't hear the start.
He says, why is population almost always a big part of these discussions when heritability can only occur at one reproductive event at a time?
Because all organisms exist in populations. And in order for evolutionary change to be permanent, novel variations have to go to fixation within a population. Okay, so there are genetic regulatory elements that are fixed within Homo sapiens that make us what we are, and they had to, under any evolutionary hypothesis, arise at some point in an individual as a novel change, and then spread. So there's no getting around the role of population genetics in understanding evolutionary change. I haven't addressed it here because it complicates the story, but it's a necessary part of any evolutionary account.
Absolutely. And anyone else want to ask a question? I'll give people a few minutes to formulate questions. That was really fascinating, though. I hadn't seen that topic discussed in as much depth before, so it was really, it got me thinking quite a few areas. It was really helpful.
Yeah. Unfortunately, the idea when it was first proposed was in such poor shape that it never really was able to stand on two legs. And I think the early criticism was well-deserved. It didn't work. But having said that, the phenomenon of, or the pattern of ontogenetic depth is nonetheless quite real. Mm-hmm. And as I'll discuss more in part 2, one of the reasons that the problem of the origin of animal body plans remains unsolved is that to build something that will show up as a fossil, right? And C. elegans is quite small with respect to a trilobite. Something that will show up as a fossil is going to require significant amount of ontogenetic depth to be present at all in the history of life.
Yeah.
And until you can find a way to build developmental trajectories using undirected evolutionary processes, that problem, namely the origin of animal body plans, is going to remain unsolved.
Mm-hmm. Yep. Also, there's the obvious problem of metamorphosis, which is, the problem compounded, right? Because you have two.
Oh, that's ontogenetic depth on steroids.
Yes, absolutely.
Or burning jet fuel. It's yeah. Barbara McClintock said metamorphosis was amazing because you have, for instance, in butterflies, really two very different organisms sharing a single genome. So metamorphosis really takes it to another level.
Yeah. Adam Mello says, I still don't understand why evolutionists could not just assert that the information for the specific cell types evolved downstream of organisms developing simpler variations in cell types as they became multicellular organisms.
Could you read the question one more time? I'm sorry, I'm having a little difficulty hearing you.
Oh, I'm sorry. Let me move the microphone closer. Says, I still don't understand why an evolutionist could not just assert that the information for the specific cell types evolved downstream of organisms.
Evolved, and what was the next word?
Evolved downstream of organisms developed.
Downstream?
Yeah, you can see it in the chat. If you open the chat window there, you can see.
Well, here's a place where I need to go back to my...
Right, sure, go ahead.
I'm sorry?
Yes, go ahead, I said.
Just give me one sec here. I'm going to go back to this toy example because I think it helps with the question. In order for a variation to be passed on to the next generation, remember that's what we need for evolution, that variation has to be able to pass through whatever the reproductive cell type is that's going to be actually going in a sexually reproducing animal, going on to make the next round, okay? What that means is that if you have a variation that occurs in the somatic cells of an animal, so I have, let's say, a mutation in one of the cells in my left arm. Unless I can find a way, and I can't, as far as we know, there is no such mechanism, unless I can find a way to get that novel information down to my gametes and into the next generation, it's lost. It will be lost to evolution. So there's no sense in which a variation, I think the term was downstream, can possibly play a role in evolutionary change unless that variation is present in this little toy example. Here's our gamete here, unless it's present here.
Right.
In order for it to be present there, it's going to have to arise here, right? It's going to have to arise in the lineage that gives rise, or excuse me, along this pathway, in the lineage that gives rise to the gametes. Not any variation will do. It's got to be variation that's going to make it into the next generation. I hope that helps the Adam, I think his name was, with his question.
Awesome. Will McCarroll asks, speciation is a favorite response in defense of evolutionary theory. How does ontogenetic depth relate to a discussion on speciation?
Well, I think speciation occurs. I think we have many striking examples of it, in fact. But if you think about what speciation is, it's the formation irreversibly of reproductively isolated groups. I'll have to wave my hands a bit here, but you are forming groups at the same, along or across the same plane of complexity.
Right.
So speciation, let's say, in grasshoppers, or speciation in whales, will be speciation of a type of organism that is now reproductively isolated from its progenitor species. But if evolution were simply speciation, we wouldn't have the appearance of novelty. You wouldn't get beyond trillions and trillions of species of prokaryotes or microbial groups. Again, moving across the same plane of complexity. So in a sense, speciation, or the formation of reproductively isolated groups, is orthogonal to the problem I'm describing. Evolution is not the same thing as speciation. Speciation may be necessary for evolution, but it is not sufficient.
Yep. Any other questions? Otherwise, we will wrap up. We got a hand raised from Rob in the audience. We have Rob. Hello, Rob.
Hey, Jonathan. How are you?
I'm very well. How are you?
Good. All good here.
Go ahead.
Just a comment to Paul. Hi, Paul.
Hi.
Just a comment. Every time I listen to these sort of talks, I'm struck by how similar the biology is to programming on a computer in an object-oriented programming language, such as, say, Java or C# or C++ or Python or lots of other programming languages. It seems to me there's a strong similarity, and I wonder if you've investigated that at all.
Actually, I'll be talking a lot more about that in part 2. Eric Davidson at Caltech and his collaborators have worked out methods of analysis of what they call developmental gene regulatory networks that structurally have strong parallels to logic gates. In fact, the terms that they use when they articulate or lay out these regulatory networks are switching terms of and, or, not. They are terms of logic that you would see in software. And we could spend a whole session just on that aspect of the control of the genetic regulation of development. I will say more about it. I do think there are strong parallels. I didn't have time to talk about them today because I wanted to get this basic description out. But next time, the topic is so rich that frankly I could do a third session if anyone would show up. But I will say more about that, and I think you were right. There are very strong parallels between computer regulatory logic and the regulatory logic that we see in any developing animal. They're quite remarkable, in fact.
Yeah. I'm just thinking specifically, Paul, of the idea of an object in a programming language. You define an object and it has a whole lot of properties to it. It's like a cell, in a sense. You define an object as a sort of encapsulated piece of code, and then the wording in a programming language is you instantiate that object. So basically you're taking a cell and you're making multiple copies of it, or you take an object and you instantiate multiple copies of it. And the information in those daughter objects, if you like, is a copy of that super object.
Oh, I have to be honest, I wish I knew more about the computer side of what you're describing, but just from your brief description now, it makes perfect sense to me.
Yeah.
I think I'm sorry, go ahead.
I think the reason, Paul, that people have moved so much toward object-oriented programming, people originally did not program in object-oriented languages. They didn't exist. But it became a necessity because of the messiness of writing software in a non-object-oriented way. So if you think about nature, if you want to develop these very, very complicated programs, as in very complicated body plans, or you want to develop very, very complicated software, you're actually forced in a way to work in this clean cell or object type method to actually break everything up into clean, encapsulated, or it's got a cell wall around it, or it's got a border around the software object. I think it'd be helpful if somebody would come on and talk about this. I'm not an expert in this by any means, but the parallels are quite astonishing.
Actually, one of my collaborators at Discovery Institute is working on a really interesting project exploring the parallels logically and structurally between how software is written, how software is put together, and what we see in the genomes of animals. So I'm hoping within the next half year, if not sooner, this publication will be out. And I think it's going to kickstart a great deal of creative theorizing along the lines that you suggested. What I find go ahead, I'm listening.
No, I just said right. You keep going, Paul.
Oh, I was going to say that I do think there are deep functional parallels in complex systems that apply from organisms to our artifacts, like software, or the structural and functional hierarchies, for instance, in my MacBook here. And we can go back the other direction. So one of the groups I'm involved with at Discovery is actually an engineering group where engineers and biologists are collaborating very fruitfully, exploring these parallels among multiple systems. It's actually one of the most attractive aspects of design theory, that you can get insights from our rather crude artifacts, take them to biology, and then come back the other direction. So I thank you very much for your comment.
Yeah, that's exactly really what I'm getting at, is I think you can draw upon the software world and all the tools that are there and that sort of way of thinking, because that thinking seems to map so beautifully onto my naive understanding of biology. Okay, thanks.
Thank you.
Thank you, Rob. Any further questions from anyone? If not, we'll wrap up next minute or so. There's no more questions. See if anyone posted on Facebook. Question. Well, people are commenting about how helpful your presentation was, so both on Facebook and on the chat here. So I think probably.
Well, I hope somebody will come back for part 2, because the Evo Devo backstory on this is absolutely fascinating. And again, because of time constraints, I wasn't able to put it in here. But the biology content will zoom up next time. Once I've got these basic issues out, we can talk about systems other than C. elegans.
Awesome.
Zebrafish, Xenopus, and so forth, but also some of these new technologies that have made possible aspects of this question, such as tracing cell lineages, that I was not able to even speculate about in 2003, 2004. Thank God science doesn't stand still.
Absolutely. Well, thanks for coming on. It's been a real honor hearing from you. It was certainly very interesting for me, and I know for many in the audience as well, given the number of indications I've had on both the Zoom and Facebook. People seem to really appreciate the talk. So thanks so much.
Good. Well, thanks for everyone who watched. And once we schedule the next session, I'll be out banging the bushes to try to get some more people to come and pay attention.
All right, great. And I'll email you when I have the video up on YouTube as well.
Thank you. Thanks to everybody.
All right, take care. Bye.
Bye now.